here is our host, Tian Wei.
Hello and welcome to a special edition of World Insight. I'm Tian Wei. COVID-19 pandemic, the biggest challenge for science community worldwide. Researchers are working around the clock on diagnosis, treatment, and vaccines, not to mention in-depth study about the virus. What about the Chinese scientists? How are they contributing to the cause, and how far, in their views, are we from the light at the end of the tunnel? With these questions in mind, I visited a lab in Beijing, which is in the forefront in the area. Let's meet the real people in the know and have science lead the way. This is the research lab of the Global Health Drug Discovery Institute, a crystallization of international cooperation as it was established by three parties: the Gates Foundation, the Beijing municipal government, and also the Tsinghua University. On top of the agenda of research here are some of the paramount international global health issues: tuberculosis, malaria, just for some examples. And recently, of course, COVID-19. Here, I met Ding Sheng, the Institute director. He showed me around and also led me to meet some of the key experts in different fields. Professor Ding, thank you so much for hosting us in this research center.
Well, thanks for having me.
So tell me more about what do you make of the challenges and opportunities as the head of this center facing a gigantic project like COVID-19?
Certainly, COVID-19 hit us pretty suddenly. Certainly, we know from the general public, from our government, and all the researchers, we need to find a cure for this disease as soon as possible. Certainly, to do this, to accomplish this in a fairly short period of time, it's very challenging. So that's why initially we really focus on drug repurposing strategy to find all drugs that can potentially treat this specific disease. As we actually move along this research and development, we now know all drugs actually may still have some specific effect or efficacy for a specific population of the patient, but certainly they're not very efficacious in treating or containing this disease.
But Professor Ding, as you may know, there are so many different directions of research, right? Ranging from test kits to the search about the virus origin and also the intermediate hosts, things like related to the nature of the virus, and also about the solutions like a therapeutic treatment or vaccines. There are so many different directions. Do you feel being stretched in all directions because there's just so many things to do?
Certainly, for tackling this specific disease or also to tackle this specific virus, there are actually multiple approaches we can take to do that, to accomplish that. We can also dissect this out in a very specific manner. For example, we can focus on a very specific antiviral approach, namely we can actually go after specific essential proteins in the viral replication cycle and actually develop drugs specifically inhibit those machineries to develop a very effective antiviral drug. But also we know the virus actually target our host, target our human cells, especially in the late stage of this disease. Our host immune system is targeted. That's why actually we're seeing the severe and the critical stage of this disease.
Sounds so fascinating. There's so many out there in the world that are doing research about directions you just mentioned and some of the others as well. Would there be redundant work? How do you communicate with one another to know that more concerted effort can be made and no efforts will be wasted in a massive way?
We actually communicate and collaborate with our international collaborators. For example, we actually just joined international anti-COVID-19 alliance, and in this alliance, actually there are multiple pharmaceutical companies, academic institutions, actually all together. Through that type of communication and collaboration, actually we know what others actually are working on. Certainly we'll be doing and contributing complementary expertise and work towards actually developing specific anti-COVID-19 drugs.
This time, the pandemic falls on a time when the world is getting even more complicated than it used to be. Geopolitics could be one of those factors. You're a scientist. You don't need to deal with it, or at least you don't want to deal with it. But how to make sure that your research could be free from the influence of geopolitics? I guess maybe that's someone, as the head of this huge research center, you have to think about from time to time.
As scientists, actually, we really focus on research. We really focus on, a common goal that's really about developing a cure for this disease. So I would say over the last 6 months, the international biopharmaceutical community and the biomedical researchers are actually all focusing on this common goal. People actually across different countries, different institutions actually are really making all the data available, working together actually towards this goal. Here at our center actually, at the very beginning actually, we created an open portal data sharing system. Basically, through this portal actually, we share actually all the data relating to antiviral research, and also all the literature references, and also our internal drug discovery results actually are shared on this portal.
Will, Professor Ding, many wonder, the research about the areas which you are focusing on be totally or strongly transformed by the research specifically looking at COVID-19?
In terms of the international support, or in terms of the recognition of significance of such potential disease, such type of emergent public health disease, I think COVID-19 really transformed our understanding, the government understanding, the general public understanding of this threat. In the past, certainly, we as scientists know about this, and people have perhaps predicted about such events. But actually, the resources and efforts actually that were put into this, it's actually not sufficient. That's why, in many ways, perhaps our international pharmaceutical societies are not that prepared for such outbreak. That's why we don't really have very efficacious drug actually that can be deployed right away.
Professor Ding, we've been hearing every day in the news, development here, advancement there. But exactly what is the reality check we should have in mind in terms of our research related to therapeutic treatment?
Yeah, certainly over the last few months, actually, there has been a tremendous new understanding about actually drug repurposing efforts. Certainly, as we all know, the drug repurposing efforts actually has some myths and hits. For example, the chloroquine didn't really show much efficacy in large double-blinded clinical trials. But we also know antiviral drugs like remdesivir actually had some specific effect in certain stage of this disease. And now actually the company is further developing an inhaled form of this drug for actually targeting the disease early on. And also we know actually very recently an old anti-inflammatory drug, dexamethasone, actually has specific benefits in actually late stage of this disease. Actually there's a significant mortality rate reduction. Certainly, we will continue actually to gain new understanding and to better actually develop those actually drug repurposing molecules towards actually specific population of patients and also in specific combinations.
But Professor Tian, as you can see, globally speaking, things are still not under control. And people already warn about the second wave. Even in Beijing, a recent spike with less than 300 cases already scares so many people. How urgent is the task vis-a-vis how basic the research still is today?
To really deal with this virus in the long run, one is really to have better therapeutics for sure that can be used to treat actually infected patients. But also actually we really need to have a vaccine for actually long-term at a society level actually to really contain.
But you know, it's only 7% before the trial. In the middle of the trial, 20% successful rate there could be. And now we are seeing things getting into the third stage trial. And that would take a long time. And we know now that even those who have had COVID-19, after being treated, still the antibody could be gone in a few months. So is a vaccine ever possible? Not to mention SARS did not even manage to have a vaccine to deal with it.
Yeah, certainly there are still a lot of unknowns. But I think overall our society or our biomedical research community are still hopeful for the vaccine. What we know is really, as you mentioned actually, there are quite a few different type of vaccines actually are now in late-stage trials, phase 2 and phase 3 trials. Certainly in coming months actually we'll know actually how those different vaccines actually can protect people from infection and also how those vaccines can really modulate or modify the disease severity. I think those early data will come out actually in the next few months.
How possible is it that human beings will be able to resume the way we used to have, to live our lives, to travel internationally, to interact without a mask or being 2 meters away from one another? How fast and how possible?
Yeah, I think actually traveling internationally have a country really open up is still challenging. But really zeroing this out so far is a good strategy, at least to open actually the community to some extent. We've seen that actually in China, if we really have no infection or very low rate of infection, actually having a relatively normal life actually can be possible. But really to scale this up, this reopening to normal, scale this up to globally, we really need to have a vaccine or actually have all the diseases actually under control.
But people are tired.
Right.
I guess you can understand that. Besides your hat of being a scientist, you're also one of us.
Right.
So this fatigue phenomenon will bring a lot of supporters to the other camp.
Right.
So how do you think, as a scientist, looking at the realities now?
I've certainly seen such fatigue in the society. I think what we can do, certainly we continue to focus our efforts on antiviral or on vaccine development. But also what we can do is really to work with media and also different governing bodies to better communicate the situation to the public. I think having such actually open, candid communication actually is even more important in the near future.
For COVID-19, most of the nations are struggling with having enough and efficient test kits. I met Professor Beijing Wei at the center, who tells me what's being done to fix the shortage. He has his own invention.
Let me introduce Dr. Bai from our school.
Dr. Bai, good to see you. I should say this, right? So what does he do?
Yeah, Dr. Bai actually has been developing a portable mobile nucleic acid detecting diagnostic system.
That's a long name.
It's basically,
this is the one?
This is the one. It's basically a portable nucleic acid detection diagnostic kit.
How does that work?
We call it BINUS, a box for instant nucleic acid screening.
Okay.
So it's very easy to operate, and you can see it's a hand-operated devices. So we have this here as the import for the sample input.
Yes.
So we have the sample like this. You can put inside, and it can be the clinical or the clinic sample. And after that, we have the import close.
Put it back, yeah.
Yeah, and later on we just use our thumb to operate.
What does this mean? What is inside here? What is inside here? Why this process is extremely important?
Because inside this box we have a chemistry called isothermal amplification. We invent a nested isothermal amplification to achieve a large quantity or very high sensitivity of amplification and detection.
So how to operate?
Yeah. So in this case, we need to
after loading the sample.
Right, after loading the sample, there's two amplification processes. We have to push this first.
What is this application process?
This is to introduce all the sample inside the microfluid channel to start the first amplification, and then we wait for 10 minutes to let the first amplification to finish, and then we push it down. That's a dilution process, and another push. So that's to start the second amplification.
I see.
And after 10 minutes of waiting, we push it all the way down to have the readout.
On the side.
Yes, the result will be a readout on the side because there will be a gold colored fluid, laminar fluid to show out if there's like positive result.
Okay. What is the color?
It's red color.
Red color.
So if that's the positive.
Right. If you have the red color show out, that's going to be a positive.
What's the negative?
Negative. There's actually no red color band on the specific position.
I see.
So we also have the control.
So how long altogether? 30 minutes?
Yeah, you can get it done actually in 25 minutes or 30 minutes.
Wow, that's fascinating.
I mean, I would say the biggest advantage of this small device is really you don't really need any instrument actually to do nucleic acid detection. Actually, for any other diagnostic for COVID-19 product, all require for nucleic acid detection, all require a specific instrument.
Right.
That's not actually convenient.
And I was thinking, for many people lining outside the hospital trying to get their PCR test, this is going to be a huge help, right?
Yeah.
But the one problem, accuracy is the biggest issue. What about the accuracy rate so far?
We test some standard sample. The accuracy is as good as the qPCR we have right now. So we think it's safe to tell if this this person actually being
positive.
Okay, and you mentioned that earlier you invented the acid itself, right? Why do you have to make the wheel? Because there are many others who already produce the acids for the tests.
Because this specific aim, specific application environment requires no specific machine and to support.
Okay.
And I think all the current assay on the market, they still need a PCR, qPCR assay, all you probably need some like a sample pre-treatment machine, like
sample preparation and also instrument actually to do nucleic acid detection. So this is perhaps the only device actually doesn't require any instrument and sample preparation. It's so convenient.
From the clinical sample to the answer.
Wow, that's really impressive.
Right, we integrate everything inside this box.
When is this going to be available? There will be people asking that question, I'm sure.
Yeah. So because this is actually a new technology,
right,
and we are currently working very hard to produce in large quantity.
Yeah.
There's a lot of technical detail to make a manufacturing.
From now till it is in my hand, meaning in the hands of the consumers, how long can we expect this process be?
We hope to get it done in 2 or 3 months.
Mm-hmm. Also, this is currently under review by the regulatory agency to really get this approved for personal use.
I'm sure they ask you a lot of different questions regarding that. Like what?
Like the convenience or the safety to be used in the open environment, and cross contamination.
Right.
All these questions are very, very important to the problem to be solved.
Right.
If we really want to get this one to work at home or in the airport and by the patient themselves.
Right. You know, I heard about the contamination of test kits in some countries, and that has been a huge problem. How can we, devices like this and many others, avoid that phenomenon?
Oh, that's a good point. The contamination, actually, if you do it by yourself, there's no such thing, okay, your sample will contaminate other people. Because in our case, you directly input the sample taken from your mouth inside this box, and this box, after that, this box is a fully sealed system.
I see.
Unless you try to break it, like.
For this, actually, the contamination, perhaps it's not a issue because this is actually one-time use.
Exactly.
So it's not an instrument. Actually, you have so many actually.
Right.
Process.
So there won't be the consideration of cross-contamination.
That's a great point. And another thing is, how much do you think this would reduce the costs of the overall operation of, nucleic acid tests all over the country? Now I know Beijing alone, since the spike in Beijing, 700,000 people being tested every day. That's a huge number, and it's still growing because everybody needs now a PCR test result, right? A certificate, right? So how much cost reduction do you think this could be?
I think compared to PCR, at least for the consumable, this will be a little bit more expensive because you see it's a fully integrated system, and PCR itself is just a tube and assay. What I want to emphasize is the time. The time you actually need to wait for the result come out. Right.
And also have all the people in the same place. Actually, that can be also risky.
Right. That's another risk. So in this case, if you think we can save like 2 days just waiting for the result, and the 2 day what can you do? You can do a lot of things.
Exactly.
Right.
Well, this is quite an achievement, I would say. Congratulations, Dr. Bai, and also to the center.
Yeah. We've been also doing antiviral drug discovery research. Let's see actually if we can find Dr. Tian actually in the TC Talk room. Yeah, he's actually been doing the viral host interaction study. Aiming to actually find a host target for developing antiviral small molecule drugs.
Can we go inside and say hi to him?
Absolutely.
Thank you so much.
Yeah, let's get a lab coat.
Okay, thanks.
We are in urgent need of antiviral drugs for treating COVID-19. But even before COVID-19, humankind are in a dire need of more antiviral drugs for treating, HIV, hepatitis B, or some other virus you probably never heard of, like a dengue virus, like Zika virus.
Ebola.
So we hope to identify some broad spectrum antivirals that will work not only for COVID-19 but also for other viruses. Maybe for a virus that will cause epidemic in the future even. So we study how virus enter into the human cells and how it replicates in human cells. Actually, that process is very complex. It needs help of a lot of your own genes or your own protein to work for them. So many different viruses actually utilize, or you may hijack, the similar human proteins for their own use.
Okay.
So if you can block those common use so-called host factors, you might have some idea how to make new drugs that not only block one specific virus, but many different other viruses. So that's what we do. We look for those genes, human genes, that are used by the viruses to replicate themselves.
So kill the virus with one stone, right?
Exactly, exactly.
But, you started this research by studying bat cells.
Yes.
Sort of this hypothetical origin of COVID-19.
Yeah, we want to learn from the nature. In the nature, there are animals that are better than humans at dealing with viruses. Bat is a great example. They have so many different viruses in themselves, and they actually transmit viruses into other, like human, like other animals.
And this time, COVID-19 also, the big suspect is the bat, right?
The number one suspect is bat.
So how much do we know now about the host and the intermediate host now, given your research?
Based on genomic studies, it's it's almost very certain that bat is the origin of these viruses. But for the intermediate host, there are many guesses right now. We are not sure.
So what have you discovered about the bat?
We want to know how bats can harbor or tolerate so many viruses without, having a respiratory disease in them, or even other symptoms in them. So they have figured out some magic ways to get away with the diseases, but still having virus in their body. We have many clues. It has been studied by our lab and other labs. It seems that they have a very unique so-called innate immune system, basically how they deal with many different viruses. This time what we found is, besides the antiviral system, their metabolism is also special. How they synthesize their nucleic acid or protein, the way they're building their own bodies is a little bit different from humans. One of the genes we find responsible for nucleic acid synthesis is actually expressed at a much lower level than humans. And that actually maybe contribute to a lower infection level by the viruses in bats.
So how fast can we learn from the bat?
We can use drugs to inhibit the human version of that gene, right? So that way we are mimicking the virus, how they're dealing with the bats. So basically you can use a small molecule drug to tone down the protein in your body. That has a much lower expression in bats. So this way you can actually suppress the viral replication at least we show in human cells. So this is how we get some idea. We can get some ideas to develop antiviral drugs by learning from nature, learning from other animals, in this case learning from bats.
That's
it's fascinating, huh?
That's a good learning from bats.
I guess learning curve, this is what we're talking about here.
It took us 6 years to develop the tools to study bat cells, because most people in the biomedical field, they don't study bats. So
but this is an interesting thing. I mean, you bet on bat much earlier than most of the people and the researchers. How did that come into being?
Since SARS. SARS is, as you know, is the origin of SARS virus. The old SARS, I mean. So from them, people have, biologists have already paid attention to bats to study them. But it's our lab who developed the first tool set that allow you to systematically check bat gene one at a time to answer the question, what's special about the bats that allow them to tolerate viruses? We check one gene at a time to fish out what are the key genes allowing them to deal with viruses.
How many genes have we covered already?
about 20,000 genes.
20,000.
Today they have similar number of genes as humans, but the genes have slightly different sequences or slightly different expression levels. But we still need to study. Many of the genes have human homologs.
Right.
But they may function slightly differently, or their activity levels are slightly higher or lower. That's the special property we want to learn.
Fascinating. Is there a special gene that you like the most?
Yeah, the gene I
Your
just
favorite
mentioned.
gene?
Yeah, it has got a long name called MTHFD1. It's the gene encodes an enzyme that have 3 catalytic activities, and they're responsible for making nucleic acid, making your the DNA, RNA, the building blocks of your cells. But we found that you can slightly tone down this gene without hurting the cells so much. But you greatly
using a small molecule drug.
I see.
Then you can suppress the viral replication without hurting the cells.
I like it.
So what is the latest on this small molecule drug?
Yes. So
Is this the boss checking the work?
yeah.
We're anxious to know the results.
Yeah,
Okay.
so this drug was originally from, it's a natural product, originally isolated from bacteria. That's another thing we learn from nature, right? And it's very difficult to mass produce. So we now team up with organic chemists to synthesize this drug in large quantity, so that we can test it in animal to
model.
So for example, we have an animal model of COVID-19 infection. If you can cure COVID-19 in mice,
Or in non-human primates.
or in monkeys, so that will help us to push this drug into clinical trials for eventually using for human one day, hopefully.
Besides finding out more about the nature of the virus behind COVID-19, there are 2 other tall hurdles. One is the vaccine, and the other, effective therapeutic treatment. What's the latest research on these? The scientists at the center with the answers.
Let me introduce Dr. Zhang from our school at Tsinghua University. He's been doing vaccine adjuvant development for COVID-19. This is Dr. Xu from our center. He's also been doing actually vaccine platform development as well as actually antiviral drug development.
Good to see you. So now I could ask the question, when are we going to have the vaccine? When are we going to have the therapeutic treatment?
There are probably 5 to 6 vaccine candidates in clinical trial in China, and we had seen very good results like the antibody titers. But this virus is very new to scientists. We cannot guarantee, there will be a success.
As far as I understand, there are so many different types of vaccine, right? As many as 5 different types. So which type are we looking at right now? Which is the biggest hope?
You never know. You never know. There are 5 kinds of technologies that the Chinese scientists are using in vaccine development, like the proteins, like the
Nucleic acid.
Yeah. There are at least 5, and from recent data in monkey and from human bodies, and we could see good antibody titers, but we don't know whether we still need other types of immune functions like T-cell functions. We need to watch very carefully in clinical trials.
The antibody
neutralizing antibody.
Yes, that existed in patients who had COVID-19. In a few months' time, those can disappear. So what does that mean for your research?
That's a very good question. So from the patients, the antibody titers will sometimes decrease very sharply. But one thing we need to remember, that's from a very small population of patients. We never know in other patients whether the antibody still exists there. And the second, we don't know whether these antibodies are neutralizing antibodies. We need to figure out whether these antibodies are valid antibodies against this kind of virus. If the case is true that the antibodies will decrease very sharply, we needed to find some other ways to help the vaccines to produce good enough antibodies that could exist in our body for a very long time. Also, we need to help our vaccines to produce other immune functions, like T-cell functions.
I think the key question is really how actually we can induce really strong immune response actually in people to really have a persisted, strong actually neutralizing antibody and also like a T-cell response for containing the viral infection. So I think that's why actually having a better alternative adjuvant development that can really boost vaccine effectiveness is actually so important.
This is eye-opening for me, everything you have just explained. What about from the therapeutic treatment side? How far are we from it?
Currently, most people put hope on the vaccine development and also therapeutic antibody. For the small molecule, I think it's quite a long way to develop. Probably antibody will be a little more short-term goal. Is if you for the like severe patient, if you give them neutralizing antibody, then they could the symptom will be
controlled.
I asked that to Professor Ding earlier about, how do you look at the urgency vis-a-vis the long-term solution, right? I guess that's also true for therapeutic treatment that you just mentioned.
In the current market, there are a lot of so-called using previously the drug treating the SARS, like remdesivir. They do, I think remdesivir they could decrease the viral titer in the patient, but then they could not completely eradicate the virus. So in that case, you still need new drugs, either with similar mode of action or with a different mode of action.
Now what you're doing here is pure scientific research. From here to eventually benefit the consumers, there's going to be a long process. Commercialization will be part of it. So have you ever thought about what's next?
So certainly for commercial development, there are several different strategies. For example, Dr. Zhang actually has worked with biotech companies actually to further translate their research, and also for adjuvant, actually you can also offer such adjuvant to actually multiple vaccine developers in a licensing deal, and actually that can be further developed and on the market.
And your thoughts?
Yeah, agreed. Yeah, so Xin's viewpoint, so we could either license out or just in collaboration with big pharma to develop the vaccine, then eventually move into clinical trial and then get approved.
Many, who is watching our program today, from outside China particularly, they would wonder, how are the Chinese researchers spending their time these days trying to cope with COVID-19? I guess this is an interesting trip we're having today. But for their reference, how are you working on a daily basis? What does this lab, this center mean to you?
Yeah.
In my lab, my students work from 9 a.m. to 10 p.m. from Monday.
Oh, wow.
So this is like a retreat almost today for you this afternoon.
That's true.
Yeah, certainly our scientists have been really working hard since the outbreak of this disease.
Also our students work very hard.
Putting even extra hours actually tackling this disease. We certainly hope actually to have new discoveries that actually can be shared globally to the drug development efforts to really accelerate this process altogether.
Chinese President Xi Jinping at a recent meeting have been committing once the vaccine in China have become a successful one, it will be a global public good. Now, there are a lot of things that needs to be explained from a successful vaccine to a global public good during this process. What do you think, how do you think that translation can be done?
I think once actually we can demonstrate certain vaccine is effective and also safe, certainly manufacturing capability in China it's pretty massive. I mean, I personally do feel actually once we have a good approved product, we can actually massive produce that and actually provide that actually to the international community.
How do we, on the one hand, try to contain the spread of the virus in this country and also the spikes that we witnessed in Beijing? On the other hand, really putting enough effort on the basic research. From the frontline scientist perspective, what do you think about that balance?
As a scientist, I want to say we need to focus more on basic science. So in 2003, there was a break of SARS. If we put more efforts on that kind of virus, we have already get fully prepared for this kind of pandemic in this year.
It's not just about China, it's also about global, right?
Internationally.
Yeah. And what about your take on that?
For this SARS-CoV-2 virus, there's still a lot of things we don't really understand. Like for the T-cell immunity, we don't really understand very much. Most people focus on the antibody development, even for the vaccine. The vaccine is mostly try to induce neutralizing antibody, but for the T-cell also play a very important function in the viral clearance. So we probably will, for our internally, we will try to use either immunoadjuvant, also other RLPS platform. Other platform we try to use different besides the spike protein. We also use other structural protein and non-structural protein like nucleic capsid and also polymerase. Try to induce both T-cell and also B-cell immunity. Try to understand the mechanism.
Right. So you cross the river. There are different bridges. We really need to test all these bridges, right?
Yeah. We need to find besides the antibody. We try to find other solutions to contribute to the.
And your thoughts, Professor Ding?
Yeah, certainly we need to continue the basic research. As they just mentioned, actually, there are still a lot of unknowns about this specific virus. We need to we need to better understand this virus. We need to find a better mechanism actually to tackle this virus. And beyond certainly basic research, we need to actually further advance our translational work, namely really developing a specific candidate that actually can at least go through the phase 1 clinical trial. Then actually in the next such event, actually we're more better prepared.
I really want to thank all of you for sharing all of your insights of the latest development with us, because what you have in your hand is possibly the solution to the biggest pandemic we have ever seen for generations. Thank you so much for your work.
We hope so. Thank you.
Thank you. And that really concludes our special trip to this center, one in the middle of Beijing, trying to figure out the solutions to the most important and challenging question we have today, COVID-19. I'm Tian Wei. On behalf of the team, thanks for watching, and bye for now.